Today, not only are we going to be learning how to code DNA and go through a lot of constructs to learn how they work and see the cool things they can do, many of them are available now with the plan to add more to our store regularly. And I have some truly magnificent things in the works that will be the subject of upcoming videos and are just finishing printing. So let's dive in and learn how to recode life itself. Like all coding languages, learning them is best done by starting with simple examples, and we'll build up to more complex use cases. We'll start with literally the most basic use case, making a bacteria produce one new protein, and we'll work up to building metabolic pathways, logic gates, and oscillators by the end of the video. In truth, though, there are actually a lot of ways to modify the genetics of an organism, and not all of them involve printing DNA. Before DNA printing was cheap and many genetic tools had been developed, scientists used things like atomic gardens to make their GMOs. Basically, the way this worked is that you have a specially built circular area where you can put plants, and in the center was a retractable pillar. Inside that pillar would be an extremely radioactive chunk of material that would allow you to irradiate all the plants you put around it. Once you'd irradiated them enough, you harvest their seeds and plant them and see if you get any weird plants that have new properties that you want. You then take those weird plants and breed them with normal crop strains to stabilize the gene. As you might imagine, there's nothing particularly efficient or fast about this process, but you've enjoyed the results of it even if you didn't realize it. For example, those red grapefruit you see everywhere were made this way. Normally, grapefruit didn't have that intense red color on the inside. But it's not just grapefruit, lots of crops were tweaked this way. Though for some reason, mutagenesis like this usually isn't considered genetically modified, even though you're changing literally thousands of genes randomly, for some reason this doesn't count. But mutagens are still used all the time to tweak and improve crops, and we use them in the lab as well.